It is a great pleasure to welcome you to this year's first webinar. It's a very special one because it's being held jointly by the European Mast Cell and Basophil Research Network and the International Eosinophil Society. This webinar is the first concretization of our shared project to strengthen our ties. This could be seen as a human reenactment of what our respectively favorite cell type has been doing for ages already. The webinar that's planned for today will be indeed shown to be inseparable in various disorders, and the sometimes disappointing results of clinical trials using targeted therapy do have a silver lining in that we are gaining more insight on the respective roles in disease and their crosstalk. By dedicating today's joint webinar to these interactions, we continue to fulfill our mission of facilitating exchange of ideas and information. And if you want to know more about our society, there's a link to the website that you see on the bottom left corner of this slide. Today's presentations will focus on both cell types, and they will be presented both globally and in various eosinophilic and mast cell mediated disorders. These presentations will be moderated, by Professor Francesca Levi-Schaffer and myself. And we will be taking your questions and answers as they come in through the question and answer box. This slide shows you a few housekeeping messages regarding the organization of today's webinar. You can ask questions at any time through the question and answer feature. They will be addressed to the speakers by the moderators. You will all be muted throughout the presentations. And if you have any technical difficulties, don't hesitate to log out and log back into the platform. The webinar is being recorded so that you can see it after or eventually share it with other people who have not been able to attend the meeting today. Now it's time for Francesca to introduce the European Mast Cell and Basophil Research Network.
Thank you, Florence. It is also for me a great honor to be here today with you to share this first EMBRN joint webinar on why mast cell and eosinophil belong together. This webinar is the first fruit of intense exchange of ideas between the ExCom members of our two societies. And as a researcher since many years in both the mast cells and eosinophil fields, for me is a most beautiful result of one of my scientific goals. I will expand later on in a few minutes the reason why I think mast cell and eosinophil belong together. Now I would only like for you to know who are behind the EMBRN, and I would like to stress that our name stands for European Mast Cell Basophil Research Network. Here are the people you will soon meet, Professor Joana Vitte. And I would like to say to you that even though our network is called European, since our existence started 9 years ago, we have members from all over the world, spanning from USA to Canada to Argentina to Korea to Brazil and so on and so forth. You can see this here, by the way. I mean, you don't see the whole world, but still Europe and the other countries. And in the last slide of this introduction, next slide, please, Florence. I would like to advertise our in-person meeting. Maybe you can think we are very optimistic. I don't think so. I think we are realistic. So we will have an in-person meeting in beautiful Utrecht in the Netherlands from the 11th to the 13th of July. And I hope to see all you there. And also, I'm telling you that there will be a IES EMBRN joint seminar. So please come, and you are most welcome. Florence.
Thank you very much, Francesca. So I would now like to introduce our first speaker, who's Francesca Levi-Schaffer. She is a professor of pharmacology, and she also holds a chair in immunopharmacology at the Hebrew University of Jerusalem. Francesca has been a long-standing member of the International Eosinophil Society. We have seen her regularly at our biennial meetings, and she has dedicated her research to mast cells and, more particular, mast cell eosinophil interactions. She has shown just how intimately these cells reach out to each other, as can be shown on these beautiful pictures showing the interactions and the prolongations that they extend towards each other. And they really establish very tight connections through which they communicate, they share information, and this leads to important functional and morphological changes of these cells. And Francesca's research has really shown this both very elegantly, both in vitro and in vivo, how these cells are really combining their efforts and form an allergic effector unit, which is truly Francesca's baby. And I think that she will be able to convince you with her presentation that this is a fascinating field of research. So Francesca, the floor is yours.
Thank you very much. Share screen. Actually, today I'm only give you an appetizer on the allergic effector unit, since, as I say, I am waiting for all of you in, or some of you in Utrecht. I put my pointer, okay. Okay, back one, here's the pointer. So, Paul Ehrlich, at the end of the 19th century, was a great discoverer of the three cells of our interest, the mast cells, the eosinophil, and the basophils. Of course, as you know, he didn't discover them as a picture here by electron microscopy. He only had a light microscope and he have several dyes, but in his great science, he could define these cells in tissues and sometimes also together in tissue of early fibrosis, as he mentioned the process of undealing and sometimes ending in fibrosis. Don't be shocked by my next slide. What impressed me as a young researcher was, and is still now, as these cells, as the masses, are located in nearly all the tissues of the body, connective tissue, mucosal, and ready to go because of these prominent granules, as I will show in the next slide. I decided now in this hypothetical cell-driven universe to put the muscle as a sun, but in addition, I have a eosinophil, a planet Earth, and Earth cannot exist without the sun, but also sun in a way has no function if it's not illuminated and nourishing Earth. I didn't forget our basophils, that is a beloved cell of some of the members of our society, and I put for the moment as Venus, beautiful, but still mostly unknown. As I promised you, I would show in this simple slide why my falling in love with the muscle, and this was really falling in love at first sight, because as you can see in this slide, I was dissecting a human adenoid tissue, digesting it, and as a first muscle stained with toluidine blue that I saw under the microscope, I saw this heart-shaped muscle looking at me. So perhaps also the muscle fall in love with me. It was reciprocal. So again, why is this fascination with the muscles? If we look at this simplified sketch of the muscles, again, they promise sort of to me that they could do many, many functions because of this pomegranate-like shape. They have many granules with packed, ready to go, several classes of pre-formed mediators, spanning from the mostly known histamine to basic neutral proteases such as tryptase, chymase, carboxypeptidase. They contain also metalloproteinase. These granules contain proteoglycan, heparin for the connective tissue muscle, and a lot of this mediator. And in addition, in the granules as preformed mediator, we have some cytokines such as TNF-alpha, IL-4, and VEGF and NGF. As the other cells of in the immune system, once they're activated, they can release lipid mediators from arachidonic acid and mostly PGD2 and cysteine leukotrienes, and they promptly synthesize and release an array of chemokines, cytokine, growth factor, and neuropeptides. So I look at this as a young scientist, but also an older one today, as a very potent cell, ready to go, found in all the tissues, containing various kinds of mediators. I cannot say of all of them pro-inflammatory. We don't know still of many. And moreover, as shown in the left side of the slide, the receptors. Yes, of course, muscle have Fc epsilon R1 that we all know for their prominent function, and I would say as primum movens of allergic inflammation. And they, of course, you would say they have c-KIT because the stem cell factor is their differentiation, activation, survival factor. But together with these two main receptors, we have a series of others, such as the newly found MGRPX2 that binds basic compounds and drug, toll-like receptors, cytokine receptors, protease activator receptor, anaphylotoxin receptor. Again, pointed out that muscle are not necessarily just allergy. And another point that convinced me to love and research this cell is that muscle are one of the most long-lived cells of our body. And they survive multiple activation. They resynthesize the mediators that they store in the granule and continue their activity. So muscle have been indeed, and I point out the human muscle because much more function has been attributed to mouse by using knockout mice, knock-in mouse, deficient of muscle mouse, in allergic inflammation, of course, in muscle activation syndrome, of course, but also in fighting viruses, bacteria, parasites, in causing venom inactivation, in inducing wound healing and fibrosis. And what again struck me and led me to study the eosinophils, that in many situations, and I would like to point out also in homeostasis, in physiology, muscles are near eosinophil and eosinophil are near muscle. Please note that muscle and eosinophil are resident in the intestine together, in lungs, uterus, adipose tissue, mammary gland, where perhaps they carry out homeostatic functions together. And the muscle, beside allergy, beside asthma, atopic dermatitis, allergic rhinitis, and so on, they are found in diseases with so different etiology, in rosacea, in bullous, in trypanosoma cruzi, in various kinds of cancers. Therefore, fascinating also by the eosinophils, I started some years ago, as Florence pointed kindly out, to study their interaction. And I defined, my lab defined, actually the allergic effector unit. You see here some images of a muscle, human muscle in connection with human eosinophil. And first, we studied what I call the soluble interactions. So the interaction due to release mediators, both by the muscle acting on receptor on the eosinophil and by eosinophil acting on receptor on the muscle. And they, just to quote some of them, however, I would like to say, and moreover, not however, many colleagues also found couples, important couples like that. We found, for example, the tryptase released by muscle can activate part 2 receptor and eosinophils to release pro-inflammatory cytokines. Muscle by PGD2 acting on eosinophil DP2 can induce their migration. And again, muscle by TNF-alpha activating TNF-alpha R1 receptor on eosinophil can cause their survival and activation. On the other hand, eosinophil, we found a long time ago, contain preformed stem cell factor. Eosinophil by MBP can activate muscle via binding to beta-1 integrin. And more recently, we went to the physical interaction. After we could detect this is in asthma, this is in allergic rhinitis, we could detect couples in vivo from human tissue, but also in vitro by co-culturing cord blood-derived mast cells, human skin muscle with peripheral blood eosinophil. And we could detect there is an intense, initial at least, pro-inflammatory crosstalk between a muscle CD48 and eosinophil CD244 especially. Another point I would like to make in the few minutes remaining is that not only we have this physical interactions that seems to activate even more the pro-inflammatory phenotype of both the cells, we have also shared receptor, and this is also a concept that Bruce Bochner is bringing about, and such as in his case Siglec-8, in our case CD308 and Siglec-7, and also, of course, the ligands. And why I'm pointing out this? Because it is a sign that we can target these cells together where we want to target them. I finish by telling you that my third scientific dream is to bring the allergic effector unit from basic science to drug target of muscle eosinophil interaction in patients. And it seems that also this dream is coming true because of the biological really targeting either the one cells, but influencing also the others. I would like to thank you, first of all, for your attention, and of course, my all the newer collaborators and my group. And that's all. I would like to leave the floor for questions if there are, and to say that I will be followed by my colleagues, Professor Marc Rothenberg and Professor Joana Vitte, who will illustrate in two disease situations how muscle and eosinophil talk to each other. Thank you.
Thank you, Francesca, very much. I don't see any questions in the question and answer panel. If no one has any questions, perhaps I would like just to formulate one regarding eosinophil depletion. From what you've shown, clearly these two cells are interacting very, very intimately through soluble and contact-dependent mechanisms. We now have these really specific eosinophil targeting agents out there, and I don't know, I was wondering if there has been any research showing modifications in the protein content of mast cells when patients are treated with eosinophil targeting treatments. Francesca? Yep.
Yes, I was cut out. I think there are no questions because I didn't really bring data. I just brought a concept and I was not expecting questions from the audience. I think that in this way we can leave much more floor to the questions that our colleagues might receive because they are showing data. instead of a philosophical concept or historical perspective.
Sure. So we do actually have a question or two, and I propose then that we address them later on, and perhaps you could join the panel then for answering the questions. Okay, we'll do it. Okay, we'll do it that way. So now I would like to introduce our next speaker, if I may. It's not my desktop that I want to show, it's the PowerPoint presentation. Okay. So is the PowerPoint presentation visible?
Yes.
Okay, so I would like to introduce our next speaker, Marc Rothenberg, who, as the majority of you know, is a longstanding member and now president-elect of the International Eosinophil Society. He is director of the Division of Allergy and Immunology and is also professor of pediatrics at Cincinnati Children's Hospital. He directs the Cincinnati Center for Eosinophilic Disorders and CIGR as well. Marc is known worldwide for his contributions to understanding, diagnosing, and treating eosinophilic gastrointestinal disorders, and most notably eosinophilic esophagitis, which is the topic of his webinar today. Marc, we are all ears.
I'd like to take this opportunity to very much thank the European Mast Cell and Basophil Research Network, as well as the International Eosinophil Society, for inviting me to give this seminar today. When this seminar was assigned, we thought that the title would be The Role of the Mast Cell in Eosinophilic Esophagitis. But as I started to synthesize the slides and the data, I realized the title should actually be changed to The Mast Cell Hypothesis in Eosinophilic Esophagitis, because we don't yet know the true function of the mast cell in the disease. So what I hope to do today is walk you through the data that's emerging, linking the mast cell with the disease, allowing us to come up with the hypothesis that this cell may in fact be involved in the disease. So I'd like to do to first start is to present first the acknowledgements. I want to say that this work is primarily done through the Cincinnati Center for Eosinophilic Diseases at Cincinnati Children's Hospital. It involves excellent collaborations that are very critical between GI, allergists, pathologists, research coordinators, the research laboratory, other individuals, including nutrition and research people, administrative support, as well as geneticists. Particularly want to call out the work on the mast cell through Natalie Ben Baruch, as well as our collaborators at the Brigham and Women's Hospital, Nora Barrett and Dan Dwyer. Want to also acknowledge the Consortium of Eosinophilic GI Disease Researchers, or CIGR, that is an NIH-supported rare disease network that is responsible for a lot of the research shown today, particularly with the large data sets. Also want to thank Curid, which is funding the research in my laboratory as well, and so many other critical agencies as indicated here. So I'd like to start by introducing you to the pathology seen in this disease. On the right, one can see that the esophagus, as illustrated here through an endoscopic biopsy, is a squamous epithelium composed of a thin basal layer and then differentiation into a number of layers as we approach the lumen. This is non-keratinized in humans, unlike the mouse. One can also see below the epithelium that there is, in this particular biopsy, part of the lamina propria, which is a connective tissue region. It's important to note that a biopsy that's obtained for an endoscopic procedure is only a small thickness of the total esophagus. What you can see on the left is that the patients that have this disease have a marked inflammatory response characterized by huge numbers of eosinophils in the esophageal biopsy, readily detectable with standard H&E staining, unlike the mast cell, which, as you know, needs a special stain. And there's also disorientation of the epithelium. What's interesting scientifically is that this is a reversible process, and that food antigens play a critical role, and if we take away foods from the diet, the patients will go into remission. If we take away the right ones and we add back the foods, they relapse. And this is a dynamic process that we can also model in animals. We also know that we can treat this disease with glucocorticoids as well as a proton pump inhibitor. The latter is not working through acid suppression, however, by anti-inflammatory effects. And I wanted to just illustrate to you in this particular slide that this provides not only an interesting scientific, enigma in terms of the mechanism of this, but also this provides biopsy materials in humans where we can readily assess type 2 inflammation, and particularly looking today at the role of the mast cell. Now, in order to introduce you to this disease, I wanted to update you with where we currently stand with the pathogenesis. We understand now that the disease is a complex trait. It does run in families, but it's primarily an interaction between the environment and early life exposures that are priming for the disease. However, there are genetic susceptibility regions, and I'll focus mainly on the variants of 2p23 as well as 5q22. 2p23 and 5q22, interestingly, encode for epithelial cell gene products. Not an eosinophil product, not a mast cell product, but the epithelium. 2p23 encodes for calpain-14, which is an intracellular cysteine calcium-activated intracellular protease that regulates the barrier function. It's specific to the esophagus. 5q22 encodes for TSLP, which is a product of squamous epithelium, not specific to the esophagus, but also in the skin. What's notable is as the barrier is impaired, the TSLP is liberated. This targets dendritic cells to make type 2 immunity, particularly the Th2 cells responding to the food. And we now understand that IL-13 is very critical because this drives not only the loss of the barrier, but also in direct potent activator of transcription of calpain-14 and also eotaxin-3, and this therefore provides multiple steps in which both the innate and adaptive immune system interact to orchestrate and promote this disease. We also know that the desmosome is very critical, that there are loss of function mutations in the desmosomal proteins, including desmoglein-1, desmoplankin, as well as periplankton, that lead to a barrier impairment and susceptibility to the disease, and that there are rare disease syndromes in humans where we can actually prove this through Mendelian inheritance of these loss of function variants. So a lot of the work today, what I'll be trying to convey to you is answering these key questions concerning mast cells. First, is EOE a misnomer? We call this disease eosinophilic because of the standard H&E staining and the images that I showed you earlier. However, do levels of mast cells increase, and do they compare at levels comparable to eosinophils? What indeed is the mechanism of eosinophil and mast cell expansion in the disease? What are the characteristics and heterogeneity of esophageal mast cells? What is the function of mast cells in EOE? And based on all of this, is there a rationale for an anti-mast cell therapeutic strategy for the treatment of the disease? I want to start off by introducing you to some work that we did a number of years ago, which characterizes the EOE transcriptome. And what you can see here is the results of genome-wide analysis looking at the transcriptome profiles of patients' biopsies. And one can see here that patients with the disease compared to two control groups had a marked change in the expression of genes. Each vertical line is a different patient, and each of the horizontals is a different gene. Now, if we take a look at the gene list, we're particularly struck by the findings that the #1 gene that was upregulated in the whole genome was eotaxin-3, the eosinophil chemotactic and activating factor, and we and others have shown that this is very critical for the disease process. We also saw abundant IL-13 expression, as well as signature genes for IL-13 induced responses. But what I want to draw your attention to today, even back from the original work that we did in 2006, was that even though this was an eosinophilic esophagitis, there was a marked signature for mast cells based on the transcriptome. And you can see here that in the top signature, there was mast cell gene carboxypeptidase 3, which is fairly specific to mast cells. There was also other mast cell genes, but I draw your attention to the CPA3 that was increased 13-fold in this initial work. So this led us to analyze mast cells and indeed determine that they were accumulating in the disease. This is looking at the levels of tryptase positive cells in the biopsies, and one can see here that the patient, compared to these two control groups, has an elevated level of mast cells. We can also measure mast cell degranulation, in this case simply by looking at the extracellular associated tryptase staining, and one can see here that indeed the mast cell is activated in the disease and degranulating compared to control populations. So EOE is indeed accompanied by marked mastocytosis and mast cell activation. Now, in terms of probing this at a deeper level, more recently we've looked at flow cytometric analysis of the mast cell. This is taking the biopsies, dispersing them into single cells, and performing flow cytometry. I want to draw your attention to the upper panel, which is a biopsy, which is representative of a patient with active disease. In this patient, he had 15 eosinophils per high power field, 50 eosinophils per high power field, and this is his autologous blood. I want to draw your attention to the cells based on the expression of Fc epsilon 1 and CD117 c-KIT, and one can see a readily detectable population of mast cells that is present in the biopsy. There is eosinophils also. This is back-aiding on the eosinophil population, and there's very low levels of what we think is basophils in the disease. The graph on the right quantifies these in a series of patients, and one can see here that the levels of mast cells is about 20% of the total CD45 cells in the biopsy. We can also see that mast cells and eosinophils are present at about comparable levels. And that, interestingly, even though the eosinophils by definition are gone in patients with remission, the mast cells remain in elevated levels in patients in remission. So, in summary, mast cell accumulation occurs in the esophagus of patients with EOE at levels comparable to the eosinophil. Furthermore, mast cell levels remain elevated even during disease remission. We've been able to probe mast cells by looking at the signature gene CPA3 in a series of patients, and in this data set shown in this slide, we're looking at approximately 300 patients, and we're comparing levels of mast cells based on the marker CPA3 to the eosinophil level. In the graph on the left, we're looking at the mast cell gene CPA3 versus the eosinophil gene CLC, which is tropolytin crystal, which is specific to eosinophils. And on the right, we're actually quantifying the eosinophils based on the number of cells per high power field. Please note that the dashed line is the diagnostic cutoff of 15 eosinophils per high power field. And one can see in the left graph and the right graph that there is a very strong association between mast cells and eosinophils. So the higher the eosinophil level, generally the higher the mast cell level. If you focus on the right, particularly noting that this is a log scale, one can see there is some heterogeneity in the relationship, however, and that there are some patients that have a relatively high level of mast cells compared to eosinophils, and we refer to this as mast cell high grouping. So there is a general strong correlation between levels of esophageal mast cells and eosinophils, although heterogeneity exists. Now, when we focus in on the EOE transcriptome and take a look at heterogeneity between patients, we were struck by the finding that even though all of these patients express the EOE transcriptome, there is heterogeneity, which allows us to group these patients into 3 categories based on the patterning that you see in the heat diagrams on the right. And we refer to these as endotype 1, 2, and 3. It turns out that these endotypes have phenotypic associations. Endotype 1 is more associated with a benign phenotype, and one can see the endoscopic appearance. Endotype 2 is more inflammatory, associated with these changes, which is furrowing and exudate in the esophagus, as well as overexpression of inflammatory type 2 cytokines, including TSLP and IL-4. And the final endotyping that we separated out, which is the narrow fibrostenotic endotype, which is associated with loss of epithelial cell differentiation, particularly filaggrin gene, and also loss of a tetraspan 12 protein that we recently described. What I want to draw your attention to is that endotype 2 is associated with a mast cell high characteristic, and that's shown by the overexpression of the CPA3 in endotype 2. This doesn't mean that mast cells aren't elevated in all endotypes, because they are, but there is a relative increase in the endotype 2. Now, the reason that we think that this is important is because this is a substantial population of patients in both adults and pediatrics, which is shown here again, a cohort from Seeger of about 300 patients. One can see, based on the expression of the tryptase and CPA3 genes, that the patients fall into the category, for the sake of mast cells, into mast cell high and mast cell low signatures, and that the mast cell high signatures in both populations represent approximately 1/3 of the total patients. Now, this turns out to potentially be of interest because we noted that the patients that had the mast cell high endotype compared to the mast cell low endotype had a different clinical characteristic. And in this particular set of findings, we're looking at the complaints that the patients had, particularly filling out patient-reported outcomes focused on mast cell-associated pain. And one can see here that the mast cell high patients had increasing amount of pain, both in pediatrics and adults. And although that's shown here, similar correlations with eosinophils did not reveal levels that were as striking or substantially significant, even though modest, compared to the mast cell. So we think that this data indeed does substantiate an association between mast cells and specific EOE clinical features. We've more recently focused on deciphering mast cells through single-cell sequence analysis of the esophageal biopsies. And this is a multidimensionality reduction based on UMAP clustering, where one can see the population of cells that are readily detectable and defined by transcriptome analysis on the single cell level. One can see that we've been able to identify multiple populations of epithelial cells, endothelial cells, fibroblasts, lymphocytes, and myeloid cells, and a very discrete population of mast cells. Focusing in on the mast cell signature genes, we can compare these genes of interest to the genes expressed by myeloid cells, B cells, T cells, fibroblasts, endothelium, and epithelium. I want to draw your attention to the genes of interest for today. Of course, based on signature genes and mast cells, specific proteases are prominent. Not only CPA3, tryptase, and kimase, but a series of cathepsins that are specific or at least relatively highly expressed in mast cells. Mast cells express the characteristic histamine and lipid mediators, as shown here, compared to the other populations. We also see expression of pleiotropic receptors on the mast cell, including known c-KIT, the beta common chain, Fc epsilon receptor subunits, notably high levels of expression of the aryl hydrocarbon receptor, equipping this cell to respond to diverse xenobiotic agents, as well as the receptor for ST2 or the IL-33 receptor. One also focuses in on the unique cytokines and growth factors expressed by the mast cell. One can see that mast cells express a series of relevant growth factors, including VEGF, TGF-beta, most notably IL-13 and IL-5. And when you compare these cytokines even to T cell levels, we see very substantial levels in mast cells. So mast cells are poised to generate proteases, lipid mediators, cytokines, and other mediators in response to diverse sensing receptors in the esophagus. And this includes Fc epsilon R1, aryl hydrocarbon receptor ST2, and c-KIT. When we take a look at these mast cells as a function of their further heterogeneity, it became obvious to us that there were actually at least 4 different subpopulations of mast cells. This is the UMAP focusing on the mast cells alone. And one can label these mast cells in a disease context depending upon their relative distribution in patients with different disease states. And this allowed us to name these cells in a clinically relevant manner. I want to draw your attention to this population. It's most diverse. This is a population of cells that we refer to as the proliferative mast cell. These express cell cycle genes, and I'll come back to that in a moment. These are the cells that are mast cells that are more similar to each other. This population is a population that's a resident population. This is one that is persistent, and this is one that we refer to as a transient population. And the reason that we gave them these names is shown in the graph on the right, which is that the control individuals express the resident population. This is the homeostatic mast cell. The patients with active disease express the resident mast cell, but also a transient population, as well as a persistent population, which we only see in the patients in remission, as well as the proliferating cells, which are a very small subset of the total cells. And focusing in on these four populations based on a single cell, we've been able to divide them indeed into relevant populations designated as resident, persistent, transient, and proliferative mast cells. The proliferative population is mainly prominent in the patients with active disease and remission. One can see here the staining for Ki67, positive mast cells in the tissue, as well as gene expression. Both of these show that this cell accumulates in patients with active disease, as well as patients that are in remission. And one can see here that these proliferative mast cells are mainly located in the epithelium rather than the lamina propria. This is the results of immunostaining and confocal microscopy, and this is a summary of the staining patterns. One can see here in the UMAP that the proliferative population is present and widely distributed between the persistent and the transient populations compared to the resident population, indicating that mast cell expansion is driven in part by local proliferation of the transient and persistent populations in the esophageal epithelium. Focusing in on the mast cell heterogeneity as a function of the disease state, I refer you to this data set here. In slide panel A, one can see a series of genes that are dysregulated in the patients with active disease as well as remission. One can focus in on a very dynamic set of genes that are activated in the mast cell at the single cell level, and one can compare it as well as to the remission accumulation of these genes, and interestingly, the persistence of abnormal gene expression, even though the patient's in a remission state and have a histologically normal appearing esophagus, as I showed you in the earlier slide based on the H&E staining. This data suggests that the mast cell is intrinsically abnormal, even in the absence of the eosinophil. And indeed, if you look at the panel on the right, one can see, focusing on the three populations of mast cells, resident, persistent, and transient, that the patients with active disease have alterations in all three populations of cells. It's particularly the transient and persistent populations that are markedly dysregulated, especially in patients in remission, that have these populations that still express abnormal levels of the proteases, histamine, and lipid mediators, as well as persistent expression of different cytokines, notably TGF-beta, and as well as dysregulation of other mediators that are involved in the disease, we believe. So taken together, there is, in fact, substantial heterogeneity of esophageal mast cells with a notable enrichment in disease-associated activated populations that appear to be poised to promote disease relapse even during remission. One of the findings that we had was that the IL-13 was expressed at relatively high levels, and surprisingly, in the mast cell. This was notable to us because the levels that we found appeared to be comparable to even the levels that we found in the T cell, which we thought prior to these findings was the predominant source of the IL-13, which we know is driving the disease through the results of anti-IL-13 biologics in patients. So one can see here is that the mast cells in the lamina propria indeed are IL-13 positive. These cells do not increase. The epithelium is the population that's dynamic in the disease, and that in the active patients, there is increased levels of IL-13 positive mast cells based on immunostaining. What's shown in panel B is that there is a striking correlation between the mast cell and the IL-13 based on mRNA as well, and this substantiates that there is a intimate connection between IL-13 and mast cells, and this also leads us to hypothesize that the IL-13 derived from mast cells may be quite functionally important. So taken together, I want to return to the original pathogenesis slide. And instead of just focusing on the eosinophil and the barrier, I want to draw your attention to the fact that the mast cells are also accumulating the disease, that the mast cells may be a prominent source of IL-13, not just the type 2 immune cells, but also the mast cells that are driving this disease through the dysregulation of the desmoglein, as well as the upregulation of calpain-14 through the IL-13. So returning to the key questions concerning mast cells and EOE. Is EOE a misnomer? I think I've shared information with you today to suggest that yes, it is. In fact, this disease is associated with a substantial level of mast cells that are comparable to eosinophils in answer to the second question. What is the mechanism of esophageal mast cell expansion? Well, I've shared with you at least some of the data that mast cell in situ proliferation is occurring in the disease, and that there is a population of proliferating cells that are associated with disease persistence as well as activation. And this is at least one of the mechanisms for the expansion. I've also showed you the interesting pleiotropic characteristics of mast cells, as well as their heterogeneity in the esophagus. We've also presented some compelling evidence that these cells are functionally active in the disease, not only by secreting IL-13, but also by making a number of mediators that are involved in the disease, such as the TGF-beta, as well as proteases that may have other functions as well. Also remind you of the correlation between the mast cells and the clinical features, particularly the pain. I think together, this answers the final question: is anti-mast cell therapy a rational strategy for the disease? And I think, yes, it is. And this is data that should fuel the testing of this hypothesis in humans. I'd like to close by making sure you're aware that the data presented today is mainly available through a public browser that we set up called EGIT Express that allows you to easily access the data and process it and download it to test your own hypotheses. Want to also mention that postdoctorate and other training positions are open. For anyone who might be interested, please contact me. Look forward to having some questions, and I thank you very much for your attention today.
Thank you very much, Marc, for a brilliant presentation. It was fascinating data, and it was also, by the way, a masterclass on how to make a perfect and very, very clear presentation. We have quite a few questions. I'm going to start with two combined questions from Steve Ackerman, which are very closely linked. On one hand, any differences in Siglec 8 expression in terms of mast cell heterogeneity in the different subtypes? And the second question, closely related, is can you comment on the recent failure of anti-cyclic biologics that target both mast cells and eosinophils in clinical trials in EOE in terms of symptomatology?
Thanks, Steve. Two questions that are relevant. In terms of the mast cell and Siglec 8, it doesn't strike me as one of the ones that were differential, but I can't tell you that for sure. We could take a look at that and let you know. I was struck by the fact that the mast cells are indeed Siglec 8 positive, and I can give you more information about the heterogeneity, but it wasn't one of the obvious differentiators that come to mind. In terms of the Allakos drug failing to improve the clinical symptoms, I think that that's illustrative of the complexity of type 2 inflammation in this disease, which is a very substantial disease. And it comes back to the question that we had early: is EOE a misnomer? because in that clinical trial, in both the phase 2 and the phase 3, the company has reported striking ablation of eosinophils. And it indicates that the symptoms, which are always hard to differentiate from placebo, and keep that in mind, that it's not that the patients didn't get better, it's just that the placebo patients got equally better, still had persistent symptoms, which is actually something that's often tough to actually modify in clinical trials for a number of reasons, which is probably beyond the discussion today. But it does call into question the role of non-eosinophils and whether or not the cyclic 8 is actually inhibiting mast cells substantially in these patients. I think is not yet particularly been addressed yet, but it's certainly an outstanding question. One would predict that if you did have a drug that completely eliminated eosinophils and mast cells, that there would be more benefit than one that eliminated eosinophils alone. That's all I could say right now, Steve.
Thank you. We also have a question from Francesca Levi-Schaffer. First, she said that it was a great presentation, very intriguing, the findings of the different types of mast cells in relation to disease. Her question is, have you looked at ILC2 TSLP pathways in eosinophilic esophagitis? You did mention TSLP. How about more largely ILC2?
Well, it's nice to get a question from Francesca. We go back to the original training when we both started. Actually, Francesca taught me a lot of stuff when I was a PhD student, and she and I shared the same bench pretty much for a couple of years. So
I'm unmuting myself to hear you, to hear me laughing. Always remember these times.
Yes. It's
Yes,
very
sorry.
enjoyable and formulative in reaching a lot of hypotheses and developing a friendship. In any event, in terms of the question, what was your question?
Have you looked at ILC2s and
ILC2s,
Maybe I can say, because you mentioned at the beginning, of course, the TSLP, and the TSLP has been shown to be prominent in crosstalk with ILC2, but is also produced by eosinophils, by the way, acting on muscles.
Right. TSLP is certainly in my opinion, a major driver of the disease based on the genetic data that we have, which is the TSLP locus being recurrently, associated with the disease in a specific manner that's independent of the atopic components of TSLP. As I stated, TSLP is overproduced in the esophagus. It does target a number of cells, like you said, the ILC2s, but it also activates effector cells, including mast cells, and basophils, and probably eosinophils as well. It is certainly driving the early innate immune response, we believe, in EOE, and which is upstream of a number of the effector pathways. So there is evidence to substantiate this both genetically and in preclinical models in mice, and certainly the anti-TSLP data in humans with asthma is very compelling, and it does reduce the eosinophil levels. So that's an open area that's continuing to be heavily pursued, and it's very interesting to see how TSLP is readily released from the epithelium, particularly in response to barrier impairments that we've shown. In terms of ILC2s, we haven't seen a prominent ILC2 signature, although it's been reported in some preliminary reports by others, and we're currently deeply probing that now. We think there may be a role for ILC2s, but data is very early.
Thank you, Mark. We have little time left for questions, but I'm still going to ask a few because there are quite a few. So Judah Denberg is asking if there's any biologic or clinical pathological entity in which eosinophils are abundant and mast cells not present, or vice versa, that you know of. That's a tough one.
Yeah. Hypereosinophilic syndrome that's not FIP1 PDHR alpha mediated. But other than that, I would say, we just reported data that, and you can take a look at it, it's online in Gastro. For example, eosinophilic colitis. We are not seeing a mast cell signature in that disease. That might be an example of, and that's more related to, appears to be more innate immunity than type 2 adaptive immunity. So you can take a look at that article that just came out, but I'm sure there are other examples, Judah. Thanks a lot for your keen question. Good hearing from you.
Thank you. Bruce has a question on doing biopsies after successful food elimination in eosinophilic esophagitis. Are the mast cells going away? I think you answered that question during your presentation. Maybe the persistence is just their prolonged tissue half-life compared to eosinophils. Do you have anything else to add in response to that specific question with regard to what you presented in terms of mast cell subpopulations?
Yeah, we haven't controlled, Bruce. Thanks for the question about time. Most of these studies are cross-sectional rather than longitudinal. But I think that, and there's also heterogeneity in patients. There are certain patients, and we've reported, with the bulk sequencing, that the mast cell genes are dramatically down. And certainly other people have shown that mast cells with immunostaining go down with diet therapy. And even actually there was a question earlier about mepolizumab lowers mast cells. I mean, it's probably not a direct effect, but it also lowers mast cell levels through, things that Seamus has reported. But, I think that there's heterogeneity, and I predict, like you probably imply, that there's going to be patients that are time dependently or even acutely have a suppression of mast cells depending upon the therapy, including diet, Bruce.
Thanks a lot. Last question from Ben Wright, who actually asked a few questions. I propose perhaps that, Marc, that you address these questions directly afterwards, if you can, if you have the time. So the question is, have you explored the epithelial-derived factors that potentially reprogram the mast cells? Or do you think that other immune cells are influencing mast cell activation?
Reprogramming the mast cells, well, I would say that, reprogramming, I'm not sure what you necessarily mean by that, but, the one that we're most interested in, which overlaps in part with what you're working on, is IL-33. The fact that IL-33 is an extraordinarily potent activator of both mast cells and eosinophils. And it is dramatically overproduced in certain populations of cells in EOE. And we, as we've described recently with our allergen, sensing mechanism, and the riboptosome-mediated cleavage of IL-33 we think is very critical for mast cell activation and potentially, like you might be referring to, reprogramming of mast cells. Thanks, Ben.
Okay, thank you, Marc. So I'm sharing my screen so that Francesca can introduce the next speaker. I hope you can all see it.
Can my video be unblocked, please? I cannot start my video. Okay. It is my big pleasure to introduce Professor Joana Vitte from France. She is a professor of immunology at the Montpellier University School of Medicine. She is also an associate researcher at the Desbrest Institute for Epidemiology and Public Health in Montpellier. And last but not least, she is the president of the Scientific Council of the French Society of Allergology. And this I should have said, last but not least, she is the communication officer of EMBRN. So she has several activities, and she's making them all at the best. So I am introducing a lecture that will tell us maybe the mirror image of what Marc just showed. So she's illustrating the role of eosinophils in muscle-driven disorders. So Joana, the floor is yours, please.
Hello, my name is Joana Vitte. I'm an associate professor of immunology, specializing in allergology in Marseille and Montpellier in southern France. And I'm very happy to talk about the role of eosinophils in mast cell-related disorders. First of all, I would like to present my conflicts of interest in this slide. I have a couple of activities which might be considered as a conflict of interest. Deconstruction of the topic. So first, we should talk about eosinophils and mast cells, but fortunately, Francesca and Marc have already presented the topic in great detail. So we will start with mast cell-related disorders and data which relate eosinophils to these diseases, the symptoms and biomarkers which are associated, and the possible prognostic and therapeutic implications. Of course, what is left for us to do? Mast cell-related disorders, in fact, considered as primary, secondary, and idiopathic, it makes a plethora of conditions which are not far from affecting 50% of all humans on Earth. So today we will mostly focus on primary mast cell diseases, and hopefully there will be some other opportunity for talking about other mast cell-related diseases. Among primary mast cell-related diseases, mastocytosis can be considered as a flagship. It is a hematopoietic neoplasm consisting of the clonal expansion of a common hematopoietic progenitor, and there is accumulation of neoplastic mast cells in one or more organ system. The symptoms can be very different. They can be related to mediator release, but also to organ damage. And we dissociate pre-diagnostic systemic mastocytosis, cutaneous mastocytosis, and different forms of leukemia or even mast cell sarcoma. In mastocytosis, there are different classifications which are used. Here you can see the most recent one published in late 2021 by a panel of experts and the consensus and guidelines of the scientific societies. I'd just like to address the fact that in systemic mastocytosis, which is made up of a number of categories with different prognosis, criteria for identifying the poor prognosis include blood or bone marrow eosinophilia, which is seen as a marker of myeloproliferation. And of course, in fact, this peripheral eosinophilia in mastocytosis had been described for say dozens of years as a finding associated with what was called by that time systemic mast cell disease. Eosinophilia is now clearly designated as a poor prognostic factor, thanks to the 2021 consensus and guidelines. Persistent eosinophilia is now a B finding, meaning it's a prognostic modifier already at the stage of bone marrow mastocytosis, which is one of the forms of systemic mastocytosis with the best prognosis. When it's added to other B findings, eosinophilia supports the diagnostic shift from, for example, from bone marrow mastocytosis to indolent systemic mastocytosis, and from the latter to smoldering systemic mastocytosis. And of course, we are talking about persistent peripheral eosinophilia, which is not related to a reactive process and not related to a distinct myeloid proliferation, different from the mast cell neoplasm. Because in that case, eosinophilia will be considered as eosinophilic leukemia or another eosinophilic primary disease, and in that case, systemic mastocytosis will become a systemic mastocytosis with another hematological neoplasm of even poorer prognosis. Looking at eosinophils in systemic mastocytosis, one can ask, is it associated eosinophilia, hypereosinophilia, or hypereosinophilic syndrome? In fact, the hypereosinophilic syndrome is suggested when there is both an eosinophilia higher than 1.5 giga per liter and evidence for eosinophilia-related organ damage. For instance, cardiac damage, which is a frequent finding. Here, I would like to give as an example the 1998 report of 2 cases occurring in men, 26 and 72 years old, both of them previously in good health, and who presented with severe cardiac conditions, which led to the diagnosis of hypereosinophilic syndrome and to the concomitant discovery of mastocytosis on the bone marrow biopsy, with perivascular mast cells surrounded by an intense eosinophilic reaction. In those patients, the treatment of the cardiac condition resulted in symptom improvement, normalized eosinophil counts, and good survival of the patients. As another interesting finding from our 2021 perspective, I would like to remark that in this 1998 publication, serum tryptase was not reported in any of the 2 patients. So now that we are in 2021 and we are looking at the eosinophils and mastocytosis, we have a very good paper which was published recently from the European Competence Network Registry. So more than 2,000 patients were examined, and their eosinophil counts at diagnosis was analyzed and used for clinical correlation, prognostic analysis, and so on. So the patients were categorized according to their type of mastocytosis, either cutaneous or systemic, and the different categories of each of these diseases. In fact, eosinophilia is not a very frequent finding overall, with less than 10% of mastocytosis patients displaying more than 0.5 giga per liter of eosinophils. In fact, slightly less than 7% had mild eosinophilia, and only 3% presented with hypereosinophilia. And of course, hypereosinophilia was especially frequent in systemic mastocytosis, up to 40% in systemic mastocytosis with another hematologic neoplasm, but also 17% in mast cell leukemia and 14% in advanced systemic mastocytosis. This paper reported very interesting correlations between eosinophil counts as absolute numbers at diagnosis and organomegaly, dysmyelopoiesis, the classification according to the World Health Organization, and the number of white blood cells. In fact, eosinophilia did not correlate with mediator-related symptoms, and the C findings correlation was only present in univariate analysis, in green on my page. If we now look at the prognostic value of eosinophil counts at diagnosis, we can see that the survival, the overall survival of these patients is indeed associated with the number of eosinophils, meaning that the 10-year survival of people with hypereosinophilia higher than, meaning more than 1.5 eosinophils giga eosinophils per liter is 19%, 70% for mild eosinophilia, and 88% for normal eosinophils. The same findings are were reported for progression-free survival and for event-free survival, meaning that normal eosinophil counts are a better prognostic marker than hypereosinophilia and also mild eosinophilia. Looking only at indolent systemic mastocytosis, the prognostic value was less evident. In fact, it was not significant for overall survival. It was not significant for progression-free survival, but it was significant for event-free survival. Another finding reported in this paper was that changes in eosinophil counts, defined as patients changing from normal eosinophilia to mild eosinophilia or to hyper-eosinophilia, had an unfavorable prognostic value regarding both overall survival and event-free survival, but also progression-free survival. And in fact, these changes in eosinophil counts could be further classified as increase, decrease, or a mixed evolution. And as you can see in this graph, the probability of overall survival was good until about 10 years with stable eosinophilia, but increasing eosinophilia was of better prognostic value than after 10 years of follow-up, which is quite intriguing. Another point which is worth saying is that eosinophils from mastocytosis express the mutation, the KITS D816V mutation, which is typical of mastocytosis. When we say that eosinophils express this mutation, in fact, a minority of them are clonal and express that mutation, which means that even if eosinophilia is called clonal in mastocytosis, a majority of circulating eosinophils do not display that mutation. So it's quite an interesting question to be asked. All those eosinophils showing up in mastocytosis, some of them maybe they are clonal, not reactive, and let's say ineffective. But the eosinophil armamentarium is quite an important, quite a well-developed pack of granules and proteins and cytokines and so on. And here you've got a very recent example of the products that the eosinophil can use and produce, release in order to drive an organopathy. And as you can see, all these substances are also present in eosinophils which associate with mastocytosis. So it's not really the armamentarium of an innocent bystander. Another interesting finding is that mast cells and eosinophils interact differently during mastocytosis. Here, I will illustrate my presentation with these very nice histopathologic images showing eosinophils and neutrophils, which are phagocytosed by mast cells in the bone marrow of a woman with systemic mastocytosis associated with another kind of neoplasm. And in this patient, even if hemophagocytosis can be encountered in other mast cell disorders, it usually involves mostly red blood cells. So here we don't know what the functional consequences are in this precise mastocytosis setting, but we do have some clues from in vitro experiments and from animal models. For example, early work showed that eosinophils release mediators that are internalized by mast cells during cell-to-cell contact. So this is one of the first papers on the eosinophil-mast-cell interaction topic from Francesca's laboratory, showing that the culture of mast cells and eosinophils display tight membrane connections with adherence sites, and eosinophils degranulate and release eosinophil peroxidase, which is transferred from eosinophils to mast cells, which internalize it. And these mediators are also able to activate mast cells to degranulate, and also finally to kill mast cells. So in the left panel, so you can see the activation by eosinophil cationic protein and major basic protein of human heart mast cells. The middle panel shows MBP, which induces histamine release in cord blood-derived mast cells, and the right panel shows eosinophil-related cytotoxicity exerted on mastocytoma cells. We also know that eosinophil mediators are elevated in mastocytosis patients in vivo and respond to treatment. As seen here, it's quite a, I would say, a note report from 2006 showing the eosinophil count and the eosinophil cationic protein measurements in serum. Eye-colored eosinophils in pink and ECP in blue. And as you can see, the ECP measurement is parallel to that of the eosinophil count, but it seems to be more sensitive, meaning that the ECP level is much higher relatively to the baseline than the eosinophil maximum to the eosinophil minimum. In some cases, eosinophils look like potential drivers of mast-cell-related disorders. Just one example, a case report of a woman of aged 42 who presented with a mast cell activation syndrome. She had an eosinophilic asthma for more than 10 years. In this patient, so here is the complete clinical investigation. In this patient, treatment with mepolizumab, which is an anti-IL-5 treatment for eosinophilic asthma, also resulted in symptom mast cell activation syndrome symptoms resolution and good outcome. So should it be called eosinophil-driven MCAS? For the time being, eosinophils are not a diagnostic criterion for mast cell activation syndromes, and they are not really granted too much attention in such conditions. If eosinophils do not seem to participate to mast cell-induced symptoms, that could also be because the eosinophil effects on mast cells may vary. One example is variation according to MRGPRX2 status. MRGPRX2, which is a receptor expressed mast cells and the eosinophils, is a receptor which is activated by cationic products, including the major basic protein from eosinophils and the eosinophil peroxidase. And activation of MRGPRX2 from mast cells results in mast cell degranulation. In fact, it was recently reported that the naturally occurring missense mutations in MRGPRX2 leads to a loss of function, meaning that the mast cells are unresponsive to all MRGPRX2 ligands. including, in our case, eosinophil-derived proteins. One of my last arguments, mast cells and the eosinophils are targeted together by novel biologics. I will not go into the detail of this large table displayed in small font, but I would say many of these therapeutic molecules are supposed to target eosinophils, and in fact, many of them do have a beneficial impact on mast cell diseases and mast cell-related symptoms. So I would say that we need to reconsider eosinophils, just like the recent paper on eosinophils in mastocytosis showed. It's not because a topic or a cell is overlooked for dozens of years that there will not be interesting and useful data to mine in there. We need to remember that mast cells and basophils are birds of the same feather in terms of ontogeny, physiology, and pathophysiology. Mast cell-related disorders are often accompanied by eosinophil abnormalities, and in mastocytosis, eosinophil counts at diagnosis and during follow-up have strong prognostic significance, despite inconspicuous differences on the white blood cell counts. There are also some points we need to change. Eosinophil biomarkers are underused for the diagnosis and the treatment of mast cell-related disorders. The pathophysiological contribution of eosinophils to mast cell-related diseases is still elusive. And I would only bring to your attention the last point, COVID-19 and mastocytosis. There are a couple of papers, but none of them addressed eosinophil counts, eosinophil biomarkers, despite the fact that eosinophils have been linked to COVID prognosis as early as June 2020. With this, I would like to thank you for your attention. I'd like to thank my students, some of whom are presented here, the Mast Cell and Basophil Club from France, the French Hereditary Alpha-Tryptasemia Diagnostic Center with Dr. Michel Aroth, and of course the EMBRN and its upcoming Mast Cell and Basophil meeting in the Netherlands next July.
Thank you very much, Joana, for your nice presentation. We have a question from Florence, who also thanks you for the presentation. Do you have any idea whether after treatment with IL-5 targeted therapy, you see any change in the mediator content in the eosinophils? For example,
in mast cells.
In mast cells. Sorry.
No problem.
I meant in mast cells. And I would add to what Florence is asking, if it has been tried to target both mast cell and eosinophils by anti-CD8 in the cases of systemic mastocytosis with eosinophilia. So since it is about drugs, I mix the two questions together.
Well, thank you very much, Florence and Francesca, for the two questions. Yes, in fact, regarding anti-IL-5, when people with maybe not with mastocytosis, but thinking about the case reports I just presented, the patient had tryptase levels decreasing with the anti-IL-5 treatment. So that's not exactly a direct answer as to whether the granule content is diminished or the degranulation is diminished, but there is an action on mediator release. And regarding anti-cyclic 8 therapy, there's been some data, and yes, it seems effective in some forms of mastocytosis. To my knowledge, it's effective in indolent systemic mastocytosis, and regarding other mast cell primary diseases, in some forms of chronic urticaria, and outside primary mast cell diseases, allergic conjunctivitis, and atopic keratoconjunctivitis.
Okay, thank you. We have a question by Nives Zimmerman. Do levels of eosinophils in tissue, not just blood, predict outcomes in muscle-mediated diseases?
I think this is a very interesting question. I'm afraid it has not, to my knowledge, it has not been addressed directly, and it goes with my plea about not paying attention to these companion cells, because as you saw in my presentation, and maybe some of you are aware that earlier paper about blood and marrow eosinophils in mastocytosis couldn't evidence any prognostic value, and this is probably because you really need to address the topic specifically and to choose the way of describing the abnormality. So regarding tissue sections, yes, if you talk about bone marrow, of course, higher eosinophilia is of prognostic value, but in other tissues, to my knowledge, it hasn't been really analyzed.
I see. Okay, I would like to ask if somebody else has any other specific question to Joana on her presentation. Otherwise, I would like to wrap our session by saying my conclusions from what I heard today, especially. In some situation, I believe that if muscle and eosinophil are found together, they might be still innocent, unrelated bystanders, like Joana pointed out. In others that we saw mostly today, they seem to be partners in crime. However, I still think that we should investigate more the possibility that they might be partners indeed. And I would like to remind to all of you the work carried out, I can't remember by which scientist, it was before most of us were born, that says that eosinophils come in the tissue because they have histaminase to block muscle activity. So it may be there are partners indeed, partners in good action, maybe are also there to try to resolve inflammation or to keep homeostasis. I would like to thank a lot, first of all, Florence for having allowed these meetings, to all the participants, the two outstanding speakers, and I hope again to see you in Utrecht in summer. Yes, Florence? We don't hear you. You are muted.
Thank you. Thank you, Francesca, for your concluding remarks. I mean, they really wrap up this session extremely well, and I'm not going to add anything else. I think it's really the beginning of a very nice collaboration and sharing of science, and perhaps that these few presentations have already given a few ideas to people who are involved in these two different cell types. I'd just like to take this opportunity to remind you that we usually hold biennial meetings to review new information about eosinophils and their roles in health and disease, and that we have now more or less firmly decided that the next in-person meeting for the International Eosinophil Society will be held in 2023. The exact dates and location are going to be determined very, very shortly, and they will be posted on the IES website as soon as they're available. And I'm just reminding you of that address for the website on this screen. The next webinar will be the 23rd of February, and it's dedicated to eosinophilic skin diseases. It will be organized and moderated by Dagmar Simon from Switzerland. And this final slide is just an invitation to send us an email if you are interested in our society, if you want more information, if you would like to become a member and don't know how to do so, if you want to eventually receive follow-up emails regarding information on future webinars. With this, I would like to thank you all for your attendance today. I would like to give a special warm thanks to the speakers and also to the European Mast Cell and Basophil Research Network for joining us today. It was really a great pleasure to hold this joint meeting. Have a nice day, everyone.
Thank you.
Bye-bye.
Bye-bye.